Well, Instagram is sort of, I don't think it's going to log in, but we are live on YouTube. So we can actually start. Welcome everyone to Tumor Talk. This is our weekly webinar we hold to let us feature our work from the Journal of Neuro-Oncology, who's been an awesome medium for us to showcase a lot of the authors worldwide for the neuro-oncology community. This week we have some of our colleagues here. We have Dr. Torgman and we have Dr. Jain. We're going to be talking about functional connectomics, sort of in the context of glioma patients. So, Dr. Jain, and you have the floor.
Oh, thank you. Thank you, Nitesh and Jason and JNO for inviting us.
Our pleasure to have you.
Yes, thank you. I'm just going to share some screen here. Just going to start with a few slides, just talking about very quickly about default mode network, how we do it in resting state functional MR. And something I asked my trainees always, what is transient hypofrontality? And I'm just going to talk very quickly before I give the podium to Michael, who's going to be talking mostly about the paper. And let me see if I can move my screen. Yeah, so resting state, it's basically functional MR. It's bold imaging, right? Contrast changes occurring over time, even during rest, right? So we can detect those using MRI. And it's possible to create a time series for each voxel within the brain, illustrating these changes in the bold signal over time, right? So this is kind of the time series we can create. And what the resting state functional network and the connect, how the networks work and how the connectomics works is that we're trying to create, we're looking at these different voxels in the brain, trying to match them. For example, if I show you an example, the time series collected from this voxel 1, which is from the left precentral gyrus, the hand motor area, and the voxel 2, which is from the right precentral gyrus, hand motor area, they kind of match with each other versus voxel 3, which is anterior in the right frontal lobe, right?
Dr.
Not seeing your slides.
Yeah, are you able to share your screen?
Yeah, I was, sorry.
That's okay. We'll give you a second. I think the imaging was going to be very instructive.
Oh, yes, yeah, it should be, hold on.
Oh, here we go.
Can you see my screen now?
Perfect. Much
Much
appreciated. Thank you.
No, I think I was on the wrong screen. So as I said, default mode network and transient hypofrontality, I'm just going to talk a little bit. And resting state FMR, bold imaging, we can detect these changes, contrast changes occurring in the brain parenchyma over time, even during the rest phase, right? And it's possible to create a time series for each voxel within the brain. We're scanning, illustrating these changes in the bold signal over time, like this one. And what we do is for looking at different networks, what we do is we look at these time series created from different voxels and try to connect them, right? For example, over here, looking at the motor network, voxel 1, which is in the left precentral gyrus in the hand motor area, you can see the time series created from this voxel matches with voxel 2, which is from the right precentral gyrus hand motor area, right? Versus another voxel, which is taken from the right frontal lobe anteriorly. These two voxels time series, they do not match with this one, right? So what we're trying to do is we're looking at all the voxels in the brain and trying to connect different networks based on this matching the time series. And it's already published. This is well established now, with resting state functional MR connectivity, we can look at these different networks, sensory motor, attention, executive network, salience, default mode network, which is, I'm going to talk a little bit about that, language, auditory, and visual. So there are different networks you can look at in the brain using resting state functional MR. And as I said, I usually ask my trainees, what is transient hypofrontality? It actually means daydreaming, mind wandering, fantasizing, but in a positive manner, right? Not thinking about a lottery ticket you're going to win. But it's also known as, task unrelated thought, or the thought which occurs while an individual is performing a task and which is unrelated to the task at hand. For example, we already know about runner's high, people who get high with, yoga, meditation, relax with those techniques. And these are all examples of, transient hypofrontality and mind wandering to some degree. The reason I'm talking about this is because this is a trait which is much more commonly seen in creative individuals, people who are artists, whether singers, musicians, painters, any kind of artists. You see this kind of trait, this kind of mind wandering and daydreaming or transient hypofrontality a little bit more. Now, how to connect it with science and resting state functional MR? We're going to be talking about default mode network in the paper also, and I just want to give you a little brief about default mode network, one of the more commonly accepted and also research networks with resting state functional MR. You can see there are different regions in the default mode network which connect with each other. The common, the major ones being the posterior cingulum, the precuneus, and the ventromedial prefrontal cortex, the dorsomedial prefrontal cortex, also the lateral parietal and occipital cortices, lateral temporal cortex, and also part of the hippocampal and parahippocampal gyrus, right? These are all the regions of the brain which connect with the default mode network. And this is an example taken from Wikipedia, looking at all the even the fiber tracks, connecting these networks. But the beauty is that it's best known to being active when a person is not focused on the outside world and the brain is at wakeful rest, right? And that's when, kind of connecting with the daydreaming component, mind wandering component. So what I'm trying to say over here is that when we are doing daydreaming or mind wandering in that particular phase of your, attention span, your default mode network is actually much more active, right? And what happens, and there is literature to suggest that based on the resting state network. But what I'm going to also mention over here, that default mode network, which is very active when the patient is when the person is at rest, it becomes less active when we're trying to do any attention demanding or externally directed kind of cognitive task. For example, I'm asking you something and you're answering my question, or you're responding to an email or something or whatever, where you're kind of paying attention to the task. That's where the default mode network is kind of shut down, right? And this decrease in default mode network activity during performance of an active externally directed task is also known as task induced deactivation, right? It's basically thought to free up resources required to respond to the external demands. And that's why the default mode network is kind of shut down, right? And what's the importance of that? The reason I'm mentioning all this, because when you're trying to do externally directed these very driven tasks, you're not letting your default mode network work, and your creativity kind of goes down, right? But it's also known that in older individuals, there's a reduced level of this task-induced deactivation of the default mode network, leading to poor cognitive performance, right? And this similar reduced task-induced deactivation of default mode network, not just seen in older individuals, but patients with Alzheimer's disease and any patient who shows any decreased white matter connectivity due to a disease process, right? So where does it fit with the paper? And now I'm going to let Michael talk about the paper. And this is where we're kind of going, these brain tumor patients, they have decreased white matter connectivity. And they have, there is less task-induced deactivation of this default mode network in these patients, kind of hence leading to similarly what we see clinically is poor cognitive performance in these patients, right? And this is what we can actually study with resting state functional MR, and that's what we try to do in this paper. But just to give for the trainees, if anybody listening, these are three things which are very strongly needed for a scientist and an artist. For example, Leonardo da Vinci, you have to be very curious, you have to observe attentively, sustained concentration is needed, but more importantly, also have to indulge in fantasizing, right? Daydreaming, and let your default mode network be really active, right? And that's kind of missing in today's world because, we're always on emails, phone calls, and text messages and doing something, right? But with that, I'm going to let Michael take over and discuss the paper because it kind of gives you a little idea about what we're trying to do with the paper is looking at this default mode network and how it the results are gonna be important in brain tumor patients.
Okay.
Thank you for
Is
setting
everyone?
the stage. The
You're welcome.
stage is all yours now.
Are you seeing my screen? Is everything okay?
Yes, it's coming through fine.
Okay.
Yeah, it looks great.
Okay.
Thank you.
So I will say a brief word about our paper. So as Rajan said, the functional imaging is based on blood oxygen level dependent sequences. And so instead of task-based functional MRI, we performed in this study resting state functional MRI. So the resting state networks, Rajan already talked about it. So the most important one is the default mode network, which is a task negative network. So I will go very quick in this one. It is involved in various diseases, including brain tumors and gliomas. So in our studies, our objective was to assess the disruption of the functional cognitivity of the default mode network, but also two other networks, the dorsal attention network and the frontoparietal networks, in patients with glial tumors. So to perform this study, we used a 3T MRI, and we recorded resting state imaging using 2D EPI sequence. It was a duration of about 8 minutes. And we pre-processed and processed the data using MATLAB and a functional toolbox called CONE. It is really easy to use, even for people that are not experts in MATLAB and everything in coding. So it was really useful. We did a lot of pre-processing. Everything is done automatically in CONE with slice timing, correction of the images, detection of outliers, normalization, functional and structural normalization, segmentation, and also some steps of denoising to remove the noise from the white matter, gray matter, and the CSF. We use two types of analysis in this paper, the seed-based connectivity analysis, which is based on regions that are correlating with the activity in a seed. And we computed the time between the time series of the seed with the rest of the brain to see which region of the brain are correlated between each other. We also use another technique called independent component analysis, which is a multivariate decomposition to separate the ball signal into independent functional components. And these components are often networks. They can be also noise, but we are looking into the components that correspond to networks and which are temporally correlated. For this patient, it was treatment-naive patients with glial tumors that were prospectively recruited from 2015 to 2019. And we used also match controls from the 1000 functional connectome project. We used both seed-based connectivity analysis and independent component analysis. And we used different number of components to study reliably these three networks of interest. We also correlated with the pathological status of the tumor, and in particular, the IDH1R132H mutation status. We included 35 patients with gliomas and 70 match controls, so 2 controls per patient. 24 of these tumors were in the left hemisphere and 11 in the right hemisphere, with a mean size of 3.4 centimeters, and 12 tumors had the IDH mutation. We found for the default mode network a global increase in the default mode network connectivity that was found with seed-based connectivity analysis, but also with independent component analysis. So with 2 independent techniques, we found the same results, a global increase in the DMN connectivity in patients compared to control. But we also found a small area of decreased connectivity in the corpus callosum, in particular in high-grade glial tumor patients. So you can see the global increased connectivity in the brain, but also a small area of decreased connectivity in the corpus callosum. When we studied the dorsal attention network, we found areas of increased connectivity in occipital region and an area of decreased connectivity in the subcallosum. And when we looked at the frontoparietal network, we found increased connectivity in the precuneus, in the posterior cingulate gyrus, and in frontal cortex, which are some areas that are found in the default mode network also. So you can see this is when we compared patient and control when studying the dorsal attention network, the decreased connectivity in blue, increased connectivity in red. And this is when we studied the frontoparietal network with the increased connectivity. However, when we compared low-grade tumors with high-grade tumors, not with control, only the two groups, we found no differences. And the same thing when we compared patients with IDH mutation and patients with no IDH mutation. And when we, for both seed-based connectivity analysis and independent component analysis, we didn't found any difference between tumors from the right hemisphere and tumors from the left hemisphere. So first, the resting state is commonly used to study networks in the gray matter, but you can also use it to detect interaction between networks in the white matter, as Rajan talked about it earlier. These white matter networks correlate with the functional gray matter networks, and they include interhemispheric commissural bridges that are traversing through the corpus callosum. So that could explain why we found the decreased connectivity in the corpus callosum in our study, in particular in high-grade tumor patients, while there was no glioma that was infiltrating the corpus callosum. So maybe the decreased connectivity between the cerebral hemisphere is due to the disruption of the connections. The hypothesis to explain why we found increased connectivity could also be based on the neuron-glioma interaction, which is bidirectional and which impacts the tumor progression. So the gliomas may increase neuronal activity, while the peritumoral neurons may stimulate tumor cell proliferation and exacerbate tumoral progression. That could be also an explanation of why we found this increased functional connectivity in these glial tumor patients. The limitations of our study was a small number of patients, that we used control with different protocol of acquisition compared to our patients. There is also debate about eye closed versus eye opened in the resting state. So we use eye closed in our study, but it could have been different maybe if we use eye open. Also, there are some anatomic distortion in the seed-based connectivity analysis, but this was controlled because we also use independent component analysis and we found the same results. And there was a heterogeneous group of glial tumors with different grades and type of tumors. In conclusion, the disruption of the functional connectivity is reliably found with seed-based connectivity analysis and independent component analysis. It's the three networks of interest, in particular in the deformer network, in patients with glial tumors, and it could be explained by the decreased connectivity between the cerebral hemisphere across the corpus callosum due to the disruption of the connections. These are some interesting articles in the subject. Thank you very much.
Thank you. That was phenomenal. I mean, it's very interesting. Thinking about this, I was actually I did an awake craniotomy this morning for a recurrent gliosarcoma case, and what I noticed was the patient had come into us, and you couldn't really notice any sort of obvious deficits or anything like that, or any sort of abnormalities in his personality. And it's pretty extensive, just based on neurological examination on him. And then the neurocog evaluation, though, showed that on a neurocognitive scale, he had some significant deficits, in different areas. So, and I think from a connectomic standpoint, I mean, it's just kind of amazing how this rewiring happening. And it seems like, from what you've talked about here, the rewiring is not necessarily just a natural sort of response to the brain, but almost as if the tumor pathology is almost hijacking it to spread, perhaps even. It's pretty interesting.
Yes, absolutely. And I think this is an area of very active interest for a lot of neuroscientists all over the world, resting state functional MR, where you can look at the brain connectomes and without any cooperation needed from the patient. Patient doesn't need to do any kind of task in the scanner. And based on that, you can look at different networks and different things. And for us, I think the obvious thing, because these patients, brain tumor patients, they have very poor performance scale, especially in high-grade gliomas, right? And it's important to look at how these networks are connected. And of course, the next step for us is, looking at prognostic information from some of these. And that's, because of the small sample size, we could not do that. But that's a kind of an expected step. Next step is to look at the prognostic information. If patients with tumors which show more disruption of these networks, do they do poorly, based on their tumor biology and behavior, versus the ones which disrupt the network less, right? So that's something I think an additional area of interest to us and many researchers, and would like to probably continue working on that.
I think it's interesting. I mean, the fact that this is a sort of what you guys are describing is a quantifiable, almost a metric kind of measure, which could strongly correlate to the neurocog measurements, which are more qualitative, of course. Despite the numerous scales and stuff, I think just a far more comprehensive 360 picture of what happens in our brains in this pathological state.
But I was impressed when I read the paper that the framework, as you alluded to, Dr. Jain, earlier, and is not just has implications in terms of gliomas, but it has implications in terms of so much of disease states in neuroscience, and it can be age related. I have a sister who's involved with and works for the American Alzheimer's Association, so that just tremendous implications there. I was a little bit remote from Journal of Neuro-Oncology, but as I read that, I thought, goodness, this platform could be launched upon so many things. And I would submit to you not only for prognostic implications, like you've indicated, and I think it's a natural next step, but then also to look at the effects of both unintended adverse effects, as well as potentially the restorative effects of some of the interventions that we do for these underlying disease states, be it neuro-oncological processes or other neurodegenerative disorders or stroke, for instance. And I think all those things have the ability to use the platform you've created and demonstrated so nicely here in the context of gliomas, and be expanded upon in so many other conditions. Really marvelous work.
Yeah, thank you. Yeah, absolutely. Absolutely. As you said, even from a surgical point of view, looking at pre and post-op cases, trying to see if there is more disruption of the networks, post certain kind of therapy, whether it's surgery or chemoradiation for brain tumor.
Exactly.
Yeah, absolutely.
Yeah. And we know all too well, I mean, we see these white matter changes in underlying disease states, even something as simple as, and I'll throw this out, it's not necessarily related to, but can be related to tumors, but someone who develops hydrocephalus, and we see transimpedable flow and white matter changes, and these patients do have neurocognitive deficits, some subtle, some more overt, but, and then you fix the hydrocephalus. Are the connectomes, do they have some degree of plasticity or ability to restore? Because in normal pressure hydrocephalus, the classical features include some degree of memory dysfunction, which has been shown to get better when you fix those white matter changes.
Absolutely. Absolutely. I think, the possibilities are enormous for a technique like this, where you could have an intervention. We're actually proposing a study at our own institute where we're trying to look at patients who undergo gamma knife radiosurgery for trigeminal neuralgia, and trying to see if there is a baseline versus post-treatment, any change in some of these networks. We have not been able to recruit any patient yet. We're still trying to figure through the IRB and the monetary aspects of the study, but I think those things could be very useful from an interventional aspect. Looking at the post-therapy patients, pre and post-therapy.
I can't help but to think, when I was a younger resident, you see a patient come in with, let's say, a hybrid glioma diagnosis, and you see those white matter changes crossing over to the other side, and you're just so focused on the tumor pathology. You're saying, okay, it's spreading this way, but this what you guys are talking about is a bit of a frame shift. It's a perspective change, because now those white matter changes usually are looked at in the context of the tumor. We should be also looking at it in the context of how the brain's adapting to this new architecture and what that really means.
Yeah, and you're absolutely right. And these changes are different in different tumors. We published this many years back, almost 10 years back, looking at DTI changes in corpus callosum in solitary METs versus solitary glioma patients, right? And these changes are different. You know, there is a different aspect to changes occurring through the corpus callosum for a glioma versus a MET in the same location. So absolutely, you know, the gliomas, how they're affecting could be different than what a tumor like a MET, solitary MET would be doing because of the infiltration and because of the, you know, the other tumor and neuronal interactions which Michael mentioned.
Well, what wonderful work. And Michael, any last thoughts you'd like to add from your perspective? You know, it's always wonderful to see have the PI on, and then to, that when he gives credit to those who really push forward the study, and you know, seeing this multi generational type science is phenomenal.
Yeah, thank you very much. Thank you, Jan. It is very interesting to discover some new techniques like a resting state for me, and to also correlate it with some other imaging techniques like we would like to do for DTI or other kind of techniques, because we could correlate the disruption of the networks from a resting state perspective, but also from a more morphological perspective using DTI, for example. So it was really interesting.
Well, for me, it's always wonderful to see my fellows and trainees do great work like this and let them present. So kudos to both of you, and thank you so much for joining the Tumor Talk. We do try to keep this short and focused. We'll archive the talk on YouTube for on-demand access for all those who come after us, and hope to see great things from both of you in the Journal of Neuro-Oncology in the not-too-distant future. You know, really, truly exciting work and things that I think resonate well with the JNO audience. So thank you both, and pleasure to meet with you.
Thank you very much.
Thank you very much.
Right.
Thank you, guys. We'll have Tumor Talk next week, same time, same place. See you all then.
Thank you. Thanks.
Thank you.
Bye-bye.